All right, we're excited now to welcome in Chad Mirkin and Shana Kelley here to talk with us today. Now, Chad, you're the recipient of the Ralph and Adams Award. Shana, first tell us a little bit about this award and why it's significant.
Sure. So the Ralph Adams Award recognizes excellence and achievement in bioanalytical chemistry. So Chad's work on combining nanomaterials, specifically gold nanoparticles, with ultrasensitive molecular detection is exactly the kind of multidisciplinary work that the Adams Award recognizes.
Chad, how did it feel to receive this award, and what does it mean to you?
Oh, it's great. I mean, Pittcon's the place for analytical chemistry, and to be recognized with one of their awards, in this case for developing new types of constructs that allow you to measure things inside cells, is a real fantastic honor and great privilege.
Yeah, tell us a little bit about your research and what led you then to getting this award.
Well, we've been working for a long time in developing understandings of structure-function relationships in nanostructures, trying to understand how tiny particles made of lots of different types of things, but in this case DNA, interact with cells. And we discovered that when you take particles and arrange DNA on the surface of those particles, they will be recognized by cells differently than normal linear nucleic acids. And one of the things is that they're recognized and rapidly internalized. And we use that understanding then to develop constructs that could go into cells and measure what was going on in a cell. And we call these structures nanoflares, and they allow you to basically track cells based upon differences in genetic content. So we can take a sea of cells that have maybe a small population that are slightly different, let's say circulating tumor cells, treat them with these types of nanoflare architectures. And the nanoflares go into all the cells, but they only light up in the disease cells, in the circulating tumor cells. And then we can use flow cytometry to sort populations of cells and pick the needles out of the haystack in this case, and collect them in live form and then begin to study them. And that's important because it can lead to new types of diagnostics for disease, but it can also lead to an increased understanding of disease, and then ultimately a new way of treating disease. So the current methods for looking for circulating tumor cells necessitate that you actually kill the cells. In this case, you're collecting them in live form, and once you have them, you can culture them and begin to play with them and look at how they respond to different types of therapeutics, and that might lead then to new types of therapeutic interventions as well.
Wow, that's incredible. So what do you both hope that people will take away from your symposium?
I think this symposium really showcases the breadth of bioanalytical chemistry. The talks will cover new types of analytical methods, but also some of the biological applications of new methods. So there's a lot of exciting science that we'll get to hear about.
Well, I hope that people understand the pace of discovery in this area, because things are moving so rapidly. I'm not even going to talk about nanoflares. I'm going to mention them one time. Those are actually commercialized structures now. There are over 1,600 different versions of these that are being sold all over the world to enable new research applications in terms of making cellular measurements. We have a new capability that allows you to manipulate cells in different ways that I'm going to talk about in the award symposium. And I'm hoping that people at Pittcon recognize that this is not a static type of field. It's one that is not incrementally moving forward, but rapidly moving forward. Major advances on the horizon, and new capabilities are going to impact lots of different areas of science, biology, medicine.
Sounds incredible. Congratulations on your award, and thanks to both of you for stopping by.
Thank you.